Hello and welcome to our webinar today. I do have some announcements as folks get logged in for the talk today. The first is I'd love to tell you about a national continuing education needs survey that's being conducted in partnership with all of the ERCs throughout the country and also being hosted by the University of Kentucky. In an effort to continuously improve our course offerings, we're soliciting feedback on educational needs. The survey is intended for employers, HR personnel, and anyone else responsible for determining what continuing education opportunities are offered to workers and employees. So I'm going to put the link in the chat. I'm going to invite you to complete this evaluation after the webinar today, not during the webinar, but just wanted to make the announcement. So that webinar is available and we would very much appreciate that evaluation or survey is available, and we'd appreciate any feedback that you might have for us. We also have another upcoming webinar this month on Wednesday, February 23rd. It's a free webinar on tracking reemployment patterns of unemployed workers during the recovery in partnership with Till Van Watcher, PhD, in the California Labor Lab. For more, you can visit us at coeh@berkeley.edu/about-CE. And without further ado, and on behalf of education and research centers throughout the country, we're pleased to present the Industrial Hygiene webinar series. This collaborative effort is on behalf of each of the ERC's continuing education programs and aspires to provide access to current research supported through NIOSH ERC programs. So thank you so much for joining us. We appreciate you being here today. Today's webinar, Firefighter, Respiratory and Cardiovascular Health, Climate Change and Smoke Exposure, is brought to you by the New York, New Jersey Occupational Safety and Health Center and Dr. Ilias Kavouras, PhD. A few housekeeping items first. You'll be muted during this presentation. If you'd like to ask a question, please enter it into the online Q&A box. We'll save time at the end of the presentation to address as many questions as possible. This presentation is being recorded and will be made available on the COEH YouTube page. And all participants who logged in with their registration email for the full live presentation today will receive an email tomorrow with a link to the evaluation form that will qualify you for a certificate of completion worth 1 continuing education contact hour. Once the evaluation is completed, you'll be able to access and print your certificate. And at this time, it's my pleasure to welcome our presenter today. Ilias Kavouras, PhD and professor at CUNY CUNY School of Public Health. Dr. Kavouras' research lies at the interface of chemical and health sciences to understand the coupling of atmospheric pollution and human health, including the role of climate change. His research focuses on particulate matter sources and composition, development of aerosol characterization technologies, wildfire smoke emissions, geospatial receptor and inverse modeling, and cardiovascular morbidity and mortality. He has developed novel analytical protocols, geospatial analysis methods, and state-of-the-art instrumentation. It is our pleasure to have you here with us today. We're looking forward to your presentation. Thank you so much.
Thank you, Michelle. Thank you, everybody. I appreciate your time. Good afternoon, and thank you again for joining us. Actually, this presentation involves quite a lot of many ERCs. One or another way, the North Carolina ERC actually supported a small part of this study to validate one of the methodologies, and then the Deep South ERC, for which I was member for 3 years, so supported the doctoral student who worked on this topic. So it's the collective action of all ERCs that can come together in this study. So without spending more time here, let's go through the presentation. And there are over 1 million firefighters today in the United States. About 1/3 of those are career firefighters, and the other 2/3 are volunteer ones. Firefighters are exposed to a large number of physical, chemical, ergonomic, and psychological hazards while on duty, and as such, cardiovascular disease and cancer are the two primary or the predominant causes of mortality among firefighters. It seems that the career length influences mortality risk, with those being for 10 years in the workforce having up to 25% increased mortality, and those spending 20 years in the force up to 40% increased mortality. As I mentioned, firefighters are exposed to a large number of chemical hazards when they are involved in firefighting activities, both wildfires as well as structural fires. Firefighting is recognized as a Group 2 occupational hazard, potential carcinogen to humans by the International Agency for Research on Cancer. The smoke contains a large number of gases and particulate matter. Now, how these chemicals will interact and what the impacts will be on the human body depends on their physicochemical properties. Large particles, for example, will settle in the upper respiratory tract in the nasopharyngeal and thoracic regions and will be cleared by mucociliary escalator. The smaller particles and the gases will penetrate deeper into the alveolar region and they will be cleared by macrophages, react through tissues, or translocate into systemic circulation and affect distant organs in the human body. Now, in terms the occupational community or the regulatory community addresses exposures to biomass burning and smoke more like as a nuisance under the nuisance dust occupational standard. It's only the National Wildfire Coordinating Group that has established down here a standard for biomass smoke, that it is 1 milligram per cubic meter for a 12 to 14 hours permissible exposure limit. The Occupational Safety and Health Agency classifies exposure to biomass burning under the nuisance dust standard that is between 3-5 milligrams per cubic meter. So just for comparison purposes, the EPA standard for the ambient air, it's 30 for 24 hour, it's 35 microgram per cubic meter. It's about 1,000 times less. So without reviewing what we already know about biomass burning, this slide presents a summary of the key limitations why we are so limited in understanding the exposures during firefighting as well as the health responses. First, it is technically very difficult and if not impossible and dangerous for the health of and safety of both firefighters as well as the research personnel to actually monitor occupational exposures and then biological outcomes under active fire conditions. You know, you don't want to collect samples while the fire is burning next to you. Secondly, the magnitude, the scale, the frequency, and the physicochemical composition of exposures during an active fire event varies quite a lot over time and space. For those being close to the fire front, the exposures are different than those further away. It doesn't mean they're higher, it's just different. The last one important limitation is that access to information about how the target organ, that is the lung, responds to environmental stressors like inhaled smoke is limited. The only way we could obtain in the past a sample of the lung is by bronchoalveolar lavage, which is an invasive method in order to obtain the sample of the airway lining fluid. So we couldn't really assess how the lung responds to environmental stressors. As such, the efforts to understand how occupational exposures to biomass burning smoke relates to respiratory health outcomes as well as cardiovascular health outcomes was done largely using surrogates, either surrogates of exposure like the career length, as we mentioned earlier, or systemic biomarkers in blood or urine or other biospecimens that are reflective of systemic exposures and effects, not necessarily what's going on into the target organ. Recognizing those difficulties and the priority to address the health effects of firefighting firefighting, the National Institute of Occupational Safety and Health recognized that as early as back 10 years ago into the strategic plan, but it put specific emphasis in the most recent strategic plan where they aim to better understand both cardiovascular disease and reduce the prevalence of respiratory disease among public safety workers, including, of course, wildland firefighters and structural firefighters. Now, as I mentioned, it is not technically difficult, if not impossible, to collect samples under active wildfire conditions, but we have another type of fire that is its prevalence is increased in the United States for the reasons I will explain shortly, that resembles quite well what's going on with wildfires. These are the prescribed fires. Now, prescribed fires are actually wildland fires, but they are managed. And there are several reasons why prescribed fires occur. It can be to control biomass accumulation and reduce the risk of uncontrollable wildfires if they start. Another one may be for ecological purposes, to promote growth of native vegetation. Some types of vegetation respond quite well to fire. Plus, you get rid of all the grass and bushes by the ground, and there is more resources available for the trees. So previous research on comparing exposures between prescribed fires and wildfires showed that they're actually comparable and might be a little bit higher on the prescribed fires. And that can be because during prescribed fire events, the firefighters or the people or the personnel who is involved with the management of the prescribed fire actually spends quite a lot of time by the fire because they need to start it and they need to control it so it doesn't go beyond and above what the boundaries of the burn and affects the community and maybe the forest itself. So you don't want to start a wildfire through that. So prescribed fires actually may afford us a unique opportunity to study these relationships between exposures and health outcomes in a controlled and safe environment. And as I mentioned before, the prevalence of prescribed fires is increasing. This plot here shows the number and the area burned by wildland wildfires, as well as prescribed fires. As you can see, the last few years, the prevalence of prescribed fires is increasing around the country, actually, more in the West, but around Southwest, even here in Northeast, as a way to control the destructive consequences of large wildfires. So we have more prescribed burns. That means more opportunities for the scientific community to really obtain more knowledge and more insights on the chemical coupling of exposures and health impacts to firefighters. So with that in mind, we designed a study with three specific goals, aims. The first one was to characterize the personal chemical composition and find the resolution of biomass smoke, particulate matter during active fire events. And part of that was to deploy validated technologies that most of us have been using personal samplers, but also applying new monitoring methods to obtain a better spatial and temporal resolution of exposures. The second goal was to determine changes in the respiratory health response to biomass smoke. And we did that using a standard methodology to assess lung function, that is spirometry, but also we tested the potential for the exhale breath condensate as a non-invasive biological matrix to actually obtain a biological specimen that describes what's going on into the lung. And third is assess the acute changes in the cardiovascular health response, again due to exposures to PM, and that was to develop or use new monitoring methods of cardiovascular health, including heart rate, blood pressure, and arterial stiffness, and see if there is a relationship or if they're related to exposures. Let's start first with the study participants. To do this study, we partnered with the Nature Conservancy, a national environmental, non-governmental organization with a strong environmental focus. We partnered with the office down in Alabama, which they do prescribed burns in Alabama and northern Florida. And these two images here, two photographs, can show typical activities of the individuals who were involved in these prescribed fires, which includes the ignition of the fire and then holding the fire, make sure it doesn't go over. This table here summarizes the study. We monitored 16 prescribed burns and at different areas and different durations, and the size of the crew changed depending on the area that they had to cover. Ideally, as many of you would like to measure everything all the time, but this is not possible because, again, although it's controlled and manageable, and the participants were very fortunate to work with firefighters who were very eager to help us. You know, we couldn't load them with a lot of samplers and a lot of devices to measure, so we had to distribute our efforts, and also we couldn't perform all health measurements at the same time because we had to balance. So this table here shows what we did for exposures and health assessment, different types of integrated sample for chemical analysis as well as continuous, and then the first 7 we measured respiratory function, and the other 16, the other 9 we measure again respiratory function as well as cardiovascular function. As I mentioned, we partnered with the crew down in Alabama, so the locations were mostly across the Birmingham up here and Montgomery down here corridor, at the Cahaba River National Wildlife Refuge, the Roberta Case Pine Hills Preserve, that's property managed by the TNC, and then a private property down at Camp to Kabachi. So there were more of the events on the Cahaba River and the Roberta, and a few of them down here. So it were accessible from the center of our study, which was up at UAB in Birmingham. So let's go into each topic specifically, and the first goal was to characterize exposures, and for that we used different experimental setups. Let's start from the bottom up. That's 2 setups you are probably very familiar, is using personal samplers where you put it in the breathing zone and it can collect either the total particles using the simple open phase filter cassettes, or we can use the MARPL, actually this is with P here, cascade, the 8-stage cascade impactor, where we can collect particles based on their size, from the very small all the way to the large particles. And we analyze them for mass, elemental and organic carbon, polyaromatic hydrocarbons, particularly the size distribution, as well as we explore the analysis of these filters with NMR spectroscopy. NMR spectroscopy gives us a holistic picture of what is in there, where everything that is present in in the sample will generate a peak, will generate a signature. So we get to see what exactly is there. We also used a continuous particle counter. That's the Ilias 1700 that measures particles above 0.5 micrometer with light scattering, both within the fine range, PM 2.5 to 2.5, and then the coarse size range. And then at the same time, we monitor the location and the movement of firefighters with a GPS. And this picture shows where the sampler was placed. Ideally, we would like it in the front of this, but it wasn't possible, so we had to attach it in the back, and that was a acceptable deviation from the perfect experimental condition. So, and let me run through some of the results as we expected. The particle mass was up to 2 milligrams per cubic meter. You know, depending on which standard you're using, you are above the National Wildlife Coordinating Group standard, but below the OSHA standard, but still quite a lot of particles. Most of that was made out of carbonaceous aerosol, organic and elemental carbon, as expected. And the ratio of OC to OC was indicative of the mixed vegetation combustion, both flaming and smoldering conditions. Now, these two columns here describe the variability between the subjects as compared to the lead officer of the burn, which, you know, the one was at least the least involved in the burn, was kind of monitoring around. So we want to see how everybody compares to a reference point, and we chose that one because it was always the same person. So, and there are some differences and similarities here. There is overall, the exposures for inorganic carbon are kind of the same for everybody throughout the exposure period, but there were significant differences on individual by individual level. So what that the opposite is true for elemental carbon. No, actually, there was quite a big difference between among participants. What tells us this is that although for organic carbon, for example, on average, all of them, they have the same exposure. On average, if we put all groups together, there is really a huge variability from one to another. So that goes back that we might have a number, and that might be 500 micrograms per cubic meter, but it really ranges from 250 up to 1,000. So, and that may have, may actually influence the biological responses. Regarding the polyaromatic hydrocarbons here and the organic and elemental carbon, as expected, we've seen most of the organic and elemental carbon and the pyrogenic polyaromatic hydrocarbons, the heavier 5 and 6 ring, concentrated mostly in the lower particle sizes, the one that can penetrate deeper into the alveolar region. Interestingly enough, the petrogenic or the low molecular weight hydrocarbons 2 and 3 rings actually were concentrated on the larger particles. And that may have to do either with the way that this may be formed during the smoldering conditions, where it generates some of this, you don't need a lot of energy, and then it kind of creates larger particles, or it might be associated with the dust released as some of the ATVs and some of the vehicles you're moving around. This is something to be explored, but this difference here between the petrogenic and pyrogenic PAHs was kind of unexpected, and to fit the rest to move forward with that. Now, let me explain you a little bit about the NMR, the use of NMR spectroscopy in characterizing exposures. Although it cannot, it is, as mentioned before, it provides a holistic review. This is everything that there is in biomass smoke here, everything organic. So we have all the information we need to have. That doesn't mean we can identify all these compounds here. We can identify a subset of those with levoglucosan, of course, and some other anhydrides like mamosan being there, acetic acid, succinic acid, lactate, and formic acid. But what we can say here, we can, based on where the peaks appear, we can identify what is made of. And there is the alkyl group, the allylic group, oxygenated, carbon, acetylic and vinylic, and aromatic. And these are the concentrations. So now, once we know that, since we know that most of our sample contains alkyl and oxygenated hydrocarbons compounds, then the next step for us would be to design protocols, experimental protocols, to identify these compounds. The same for aryl, contains a large number of kind of considerable quantity of aromatic hydrocarbons, and then not only PAHs. So PAH is in the order of nanograms. This is kind of micrograms per cubic meter here, the terms of concentration. So there is a lot of things we don't measure if we target a specific compound, and the NMR analysis can give us direction. What are the predominant compounds, but then where else we need to focus to obtain more information about the chemical composition. Let's go to the last one, that is understanding the particle size distribution. And that gives a pattern of one firefighter during a prescribed burn. And you can see there are periods of high exposures and low exposures. And also with the blue one is the speed, how quickly they move. And the dark line is that they're going up and down. So there are areas of high exposure and low exposures. And as we will see later on, but also here is that areas of high exposure were associated with mobility when the firefighters were moving. So that means that has implications about the dose, because when you are moving and you are in some kind of physical activity, then your your breathing rate increases, and actually the effective dose may be higher. This plot here represents a special view of the previous figure. We can, with the topography of the prescribed burn, so you can see, actually this is all firefighters in this involved. And here we can see the areas of high exposure and low exposure, and where most of their firefighters been in relation to the boundary here. So this is a information that, you know, if it was linked to a online system, it can actually prevent cases of high exposure here. We can move individuals around. So maybe the first one will be exposed, but we can make the others go around this one, make sure they are not exposed at those levels. The second topic is addressing the respiratory function, and we did that by spirometry, a standardized method, but also we collect exhaled breath condensate. This is blowing into the air tube, and here is a picture of how that looked in the field, a cooled tube that was at -10 degrees, and the way it does, it cools down your breath. So then we can take that, the water extract, which contains information about what's going on into your lung, and analyze and see for differences if things change before and after exposure. This table here shows a summary of the participants who joined both the lung function and the exhale breath condensate experiments. They are similar. Some of them opted out, they didn't want to do it. And quickly about the pulmonary function, we indeed measure a decline in the FEV1 and FEVC, but it wasn't, it's minimally reduced before post-exposure as compared to pre-exposure. The biggest difference was observed for the peak expiratory flow, which is an indicator of ventilator impairment, and was reduced more as compared to the other two indicators for post-exposure as compared to pre-exposure. These two plots here shows the changes in FEV, FEVC, and PEF as compared to two exposure metrics that we constructed based on the duration of the firefight of the event or the area of the event. Not the particle number, but this is something we're currently working on. But what we found is that there is a greater change of FEV1 and FEVC with increasing burn intensity, while there is the PEF changes with increasing event duration. So it's a combination of both the intensity, the magnitude, and the duration of the exposure affect, and they don't affect the same variable. You know, it appears that the duration has a bigger effect on peak expiratory flow, while the scale, even high exposures, even for short period of time, will affect mostly FEV1 and FEVC. Let's move now to exhale breath condensate, because that was the novel component here, which we measure pre and post exposure, and actually we analyze them using the same methodology by NMR spectroscopy, like the one you use for particles. So now we can compare actually the exposures and the exhale breath condensate using exactly the same methodology. So these two spectra here are typical for pre-exposure and post-exposure, and this table here shows the relative abundance of the functional groups, pre and post-exposures. They're relatively comparable. We didn't see a big difference, except for the concentration of oxygenated hydrocarbons that declined post-exposure, and the concentration of alkyl compounds that increased post-exposure. So that tells us that there is something going on. And the smoke inhalation triggered a response. Now, you already have the paper published on this, so you can go and look how this oxidation, how these two ratios were calculated. But when we plotted them together, they kind of create two regimes. One is the reduction regime, and then there is the oxidation regime. So what this plot tells us is that it's the post minus pre, is that for some of the firefighters, for some of the individuals, exposure, inhalation of smoke will trigger an oxidative transition, while for some of the others, it will trigger a reduction regime. That is production of compounds that are reduced, like ethers, alcohols, oxidation, production of compounds that are oxidized, like carbonyl, keto, or carboxylic acids. Now, although these are different compounds, they're the two sides of the same coin. They are indicators of inflammation. Different compounds, but indicate different stages in inflammation. So this kind of analysis provides the first indication that actually lung is responding through an inflammatory pathway, and actually it responds differently by individual. The response varies by individual. So one, it will trigger a specific response, and it may trigger a different response, but all of them are inflammatory responses. So that goes back to the exposure levels and the huge variability. In terms of cardiovascular health, we used a blood pressure monitor, which the individual carried throughout the burn, and measured blood pressure every half an hour. We used a sphygmomanometer, and we measured pre and post central, peripheral, and central blood pressure as a way to calculate the augmentation index. And then we used this watch here. You see an image, you can see, oh, it's very flu here, which actually it's a more advanced version of the Fitbit, which collects information about movement, heart rate, blood pressure. It does an EKG on a frequent time. It collects temperature, skin temperature, as well as the electrodermal activity. So it provides a lot of information what's going on the physiological level. So what based on the data we collect here, our firefighters effectively walked over 10 kilometers per event over 4 or 5 hours. That's quite a distance. And actually only 10% of that was from flat surfaces. They were going up and down all the time. The terrain wasn't flat. So, and what that affects is their respiration and their activity level. In fact, by analyzing pre and post exposure blood pressure, we found that heart rate was increased post exposure, but heart rate was increased, but blood pressure was decreased. So this kind of a trend we observe, after physical activity. Keep in mind that they've been walking almost 10 kilometers up and down the hills. So there is some level of physical activity here that may seeing the impact here shortly after the exposure. This is not to say that smoke didn't do anything, but physical activity maybe exert its impact sooner or faster than smoke inhalation, because the smoke inhalation has to go from the lungs, transition into the systemic, and then trigger the response, and that may take some time. So, and actually for the augmentation index, we've seen a decline post-exposure for the augmentation index, which is a good thing. But we saw also a relationship with the levels of particle number concentration. So there is indeed probably because of the physical activity involved in this process, a better cardiovascular function shortly after the effect, but there is an association, there is a correlation with particulate particles in this level. So we're kind of having two competing activities here that actually has been studied before, whether physical activity can outweigh the effects of air pollution. And this plot here shows how the heart rate now that was monitored continuously during the event, not just before and after, but continuously during the event, is related to different types of activity based on the movement, so, and exposures as well. So when individuals were resting, were not walking or not going up and down, their exposure levels were low. So meaning they were further away from the fire, weren't exposed. And when they will start engaged into moderate or vigorous activity, that means we're engaged with the fire one or another way, their heart rate increased, but also their exposures to particulate matter increased. So this is an indication that exposures during periods of high physical activity will increase. Now we still remain to see the effect of that, which probably takes some time. The same thing, it was observed here for blood pressure, the systolic and diastolic blood pressure. It increased for periods of higher exposure shortly after yeah, it increased for periods of short exposure. You can see that over, it's not over a event of 6 hours, the blood pressure was measured every 30 minutes. It was one of these that we managed to get a complete set of measurements. So yeah, so what these results, what this study shows up here is that particle, first of all, we had the we managed to actually obtain robust and reproducible measurements, both of particle exposure as well as respiratory and cardiovascular function among firefighters who were involved in prescribed fires. So that tells us that prescribed fires, it is a good tool, it's a good approach in order to study the coupling between smoke and human health in a controlled conditions without compromising the health and the safety of individuals as well as or the quality of the measurements. Now, as we expected, we've seen particles mostly in the fine size range and varied considerable by participants at events. So although everybody was exposed at high levels, there were significant differences among them. The chemical analysis revealed, of course, polyaromatic hydrocarbons found in small particles, the ones that penetrate deeper. But also we've seen typical signatures of biomass burning in some soil organic matter, mostly the formic acid is coming from soil, into the personal exposures. The changes in lung function and exhale breath condensate revealed that actually lung is responding to acute exposures to smoke, and is responding in an individual specific way. So considering their previous status, and then their physiology, how they will respond, which pathway of inflammatory pathway will move, either leading to an oxidation environment or a reduced environment. But again, both of them indicate inflammation. Cardiovascular function changes, and actually it improved post-exposure when compared to pre-exposures, indicating that probably the benefits of physical activity involved with the participation of these events were stronger shortly after the exposure. That, as mentioned, doesn't mean that there is no effects of smoke, cardiovascular effects. It just may need to take more time, and follow-up studies will look over a period of time following the exposure, how these event changes, or after multiple events, whether blood pressure or arterial stiffness increases. Generally, changes in respiratory and cardiovascular function increased during elevated particle numbers, and that may be because of the potential for increased internal dose as exposures, as the levels of exposure to particle number concentration were higher when people were engaged into moderate or vigorous physical activity, which tends to be close to the fire front, on by the fire front. So this was a study that involved many moving pieces. We'd like to thank the firefighters of the prescribed burn crews at the Nature Conservancy. Without them, none of that would have been possible. They were the best study participants, eager to help, eager to engage, and find ways to do more than we actually asked them. So, of course, this was, I mentioned before, part of doctoral dissertation of Dr. Jordan Nelson at UAB SPH. Zoran Pavicevic helped us at the beginning of the study with the validation of the exhaled breath condensate. Marie-Cécile helped with the analysis of the NMR, is the NMR expert, which was done at the NMR facility at UAB. We have group individuals from different institutes, which they help us with the chemical analysis, and not all of that we could done that in-house. And then finally, the support by the Deep South ERC and the North Carolina ERC for this study. And with that, I conclude the presentation. Thank you very much for your attention. I think we're on time.
Yes, perfect timing. Thank you so much for that excellent presentation. At this time, you are more than welcome to enter any questions that you might have into the chat or the Q&A box. If possible, if you could enter them into the Q&A box as opposed to the chat, that makes it easier for the moderator, which is me. First, I'll just start with a couple of comments. Some folks acknowledging they're excited to be here and just thanking you for sharing this really important research. Someone also wanted to note that occupational exposure to wildfire smoke is also regulated by Title 8, Section 5141-1.
Thank you. That's good to know.
Yes, great. Well, our first question is the NWCG biomass smoke exposure limit only intended for wildfire smoke? Would it also apply to other types of fires?
I think so, but I don't know, what do you mean by other types of fires? Is it structural fires or so? I don't think so, but it should be for the wildfires, and that should include natural as well as prescribed. But I don't think it applies, I don't think it applies to structural fire. That's specifically for biomass burning. Structural fire, it involves more than biomass.
Great, thank you. And yes, they did mean structural fires.
Yeah, I saw the comments.
Oh, perfect. Great. Our next question, is it rare or not for medical surveillance programs to be available to wildland firefighters?
Not. I'm not aware of not, but if others know, please feel free to respond. I'm not aware of those in wildfire fighters. I don't know if there are any regular medical surveillance programs. And that should be my guess is that may be department specific in areas that I know here because of the World Trade Center, we have the surveillance program for the WTC firefighters, but yes, it's a local concern. I'm not sure there is something at that level probably in Western states.
Thank you so much. And I think that also touches on the structure of firefighting, right? Whether it's your full-time occupation, you're a volunteer firefighter, kind of where you
Yes.
fall within that scope. Absolutely. Thank you. And oh, we're starting to get lots of questions. Perfect. All right. Was there any indication of longer term or permanent change in respiratory function or cardiovascular function?
Yeah, that was one of the tasks. It wasn't within the scope of the study. We're looking for the acute effects. We still have the data because this study was done over a period of time. We still have a data for a few participants, not all of them participated in all events, so we can see for trends. But there is definitely the next step, which is going to help us answer also the cardiovascular question, whether acutely we may see the effect, the benefits of physical activity, but what happens long term or subacute, within a firefighting season or so. So that's something we need to follow up.
Thank you so much. Are you aware of any data that describe how any respiratory protection PPE that wildland firefighters wear influences their cardiopulmonary function?
Oh, that's the David, that's the $1 million question. So it's actually eventually, that would be this kind of was the first step. The next step would be for us is, if we were to do the same study, but some of the firefighters wear PPEs, some did not wear PPEs, and then we can compare. The other goal is usually we assess the effectiveness of PPE by how much they reduce exposure by developing metrics like the exhale breath condensate or even the lung function, or then we may be able to assess how well PPEs are working in terms of how well they improve, or not they improve, but the how less reduction on spirometry, lung function, or exhale breath condensate. So this again is follow-up studies. We hope we'll be able to do that, either a case control or compare function with and without PPE and see how it goes.
Thank you so much for sharing. Are there any use of robots or other types of automation in firefighting?
Oh, I don't know. I cannot help you with that, especially in the wildfires, the big fires. I'm not sure there is any robots or so. It's in the prescribed burns, no, it's all manual.
Thank you so much. It's a good idea, though.
Yes, and technology is there, and they can use. I know there are technologies where they now locate where the firefighters are, especially in the big fires. So and how they can move around and place them. I don't know if they are involved in robots or non-human instruments are involved in firefighting, but definitely technology is used to manage where people are.
I also think of drones as well for kind of spotting spread and those sorts of things. Not so much fighting the fire, but helping with the fighting.
Helping to see where they are, where the fire was, particularly in large fires, that they go quickly and they change direction. So
thank you. Well, this is a follow-up to the NWCG question. Does this recommended exposure limit include wildfires where structure fires also occur? For example, the wildland urban interface.
I'm not sure. I don't know. I'll have to check. I can, Michelle, if you can send me the contact information, or I can send you the document for that, and then we can forward to individuals. I think it meant only for wildfires, not when it involves structural fires. The mixture changes. The composition of the combustible material changes by a lot. So
Thank you. Yes, and also I should note for our participants here today, Dr. Kavouras sent me a few different articles to share with all of you. So we'll make sure to share some additional information after the presentation as well. And you'll get that with your evaluation emails. We have a hello from a previous student of yours at UAB, Brenda Hogan, and wanted to say thank you for a wonderful explanation on a critically important topic. I jumped on late, apologies if this was already addressed, but would love to hear your thoughts on the significance of potential contribution of dermal exposure to contaminants as a part of the overall exposure profile.
Yeah, that's a significant concern for both wildland firefighters as well as structural firefighters when they wear their uniform and then after the event, they take it out and they fold it, and then the exposed surface of the uniform can attach the human skin, and that may be a way for these not only PAHs but other contaminants to enter the human body. So that's a concern, particularly because it touches the area around the neck, which is softer, and so that's a issue across the board, not only for wildland firefighters but also for structural firefighters in an urban environment.
Thank you. Just a couple of comments as well. We have a confirmation. Yes, there are drones used for aerial ignition. So if anyone was wondering, we had a participant let us know that. And also there are a couple of studies out there using exposure models estimating lung cancer rates between 1 to 1.5 RRs among wildfire firefighters. It's an FYI, so there are some studies about that for anyone who may be interested. And we'll jump into another question here. Are you aware of recent monitoring for I'll see if I can say this word.
Carboxyhemoglobin.
Yes, thank you. Levels after.
So, no, but that's a good suggestion that we should be looking for that. That entails there is CO2 in this process. There is plenty of CO2, but I'm not aware of any of those, but that could be something that needs to be monitored as well.
Thank you. Another question, are you aware of respiratory protection suitable for firefighters to protect against the chemicals present in fire smoke?
Oh, I mean, that's again, it's a good question. It depends what you mean suitable. I mean, yes, there are devices, the respirators, the full mask and so, and with oxygen, but they don't last for long. And then, you already carry, you see this backpack that's 40 pounds of materials that I have to carry. We're going to carry something like that. Then you're only going to use it for maybe an hour or two. So it becomes it's a combination here, how much protection they can afford over the risks that you may have or convenience. Unfortunately, I mean, now there is other less effective PPEs, like compared to the full respirator, right? Like the masks that all of us are so accustomed right now that they should be using that. In our experience, and that was a study before COVID, of course, it was most of the firefighters were using the bandana in front of their face. They might, put water on that, so it helps a little bit more fresh or so. But definitely, that's something that there is PPEs out there available to firefighters. But at the end of the day, I don't think they're used as much as they should be used to protect that. Or there might be now we make them more friendly to the firefighters, so they can use them more, and because they actually work, they reduce concentrations, they do what they're supposed to do.
Thank you so much. Besides blood pressure measurements, did you have other metrics for cardiovascular function?
Yeah, we measured arterial stiffness, which is an indicator of how flexible the arteries are, or the blockage in the arteries. So, and that was also improved shortly after exposures. Again, that was a measurement we can only do before and after. Would be, we're looking if we can do it during the event, so we can see the very short acute effects, and then heart rate. And we still have the data from the EKG, although that's not, like the regular EKG you get at the doctor's office. We're finding ways to process and see whether we can extract useful information out of that. But this is an experimental device that's a new device that came up a few years ago. So we're still finding all the useful things we can get out of this.
Thank you.
Okay.
We have also a comment. Someone wanted to note that the NFPA 1984 covers wildland firefighting respirators. So for the individual who wanted to know more about respirators specific for wildland firefighting, that is one resource that was provided by another attendee as well. So thank you. One more question here. It appeared that some of the firefighters were smoking cigarettes. Does smoking status affect their monitoring data?
So individually, we try the results I showed to you. They didn't involve smokers. In terms of the analysis, also when we put all the data together, we controlled for smoking, and it didn't affect the outcomes. We did the analysis without them and with them, by controlling for smoke, and it didn't appear to affect the responses. We also ask them to be fair that they do not smoke just before they start engaged into the activities, so they try to stay. So this may not represent an actual state, natural state for them, because we ask them not to smoke for the previous day or two for that, but or at least before they do the activity. So, but the analysis we did show that it didn't affect the outcomes.
Thank you. And then we have another comment too. There's currently no regulatory mandatory standard for respiratory protection for firefighters operating in wildland or wildland urban interface fires. And Cal OSHA is developing a new regulation for these settings in 2022. So for our viewers in California, Cal OSHA is working on a regulation for these settings in 2022. So make sure you watch out for that if that is an area of interest to you. I'm sure they would love comments and feedback as they're developing their drafts. We have SOAS or OSHA. Great.
This is greatly needed, and I hope it's done in a way that makes it friendly to the firefighters. So there's so many things they are exposed to, and and that would definitely help them if they are using it properly.
Well, thank you so much again for your time today. I'd like to just close. I know so many questions have been asked on lots of different potential areas for future research. I'm curious, is there a follow-up study that you're already planning, or do you have kind of a wish list of things that you're hoping to look at?
Yeah, I kind of spelled out my wish list, responding to the questions about the different studies. The one we're trying to engage here is expand into the structural fires with urban firefighters. I mean, in New York City, we don't have a lot of wildfires, but we're still trying to working the collaborations with Jordan down at UAB and others to do the study. So as I mentioned before, using the PPEs with and without, the chronic effects, and then expanding to the urban firefighters and structural fires. Particularly, as one of the attendees mentioned, on the interface between forests and urban environments, which is unfortunately, there are more often many incidents every year, more incidents every year. So, and that can change a lot. So, and then the second thing on the larger scale is, of course, firefighters, but then how biomass burning affects everybody, the general population, because firefighters inhale that right there, but that smoke is gonna go over populated areas. In California, you you may not see the fire, but you can smell the smoke. So everybody is exposed. So we want to see also how this knowledge can be used to address environmental health, not only occupational health, but environmental health, the broader population impacts. So here in the US, but also around the world.
Yes, thank you. Yes, wildfires do not know state or country bounds, unfortunately.
Exactly.
Thank you so much for your time today and for all of these wonderful questions and the interest from everyone here joining us for the webinar. A special thank you to our presenter today, Dr. Kavouras, as well as everybody who joined. Be sure to check out our website for more information about upcoming events. That's coeh@berkeley.edu/about-CE. And as a reminder, you'll get an email from us tomorrow with a link to the evaluation. We very much appreciate any feedback you're willing to share with us. Thank you all so much for being here, and I hope you have a great rest of your afternoon.
Thank you very much. Appreciate it.
Bye-bye.
Bye-bye.